Hello, my name is Ella Brownson, and I had the pleasure of working with Dr. Shally Page based off of some research that he also conducted with a previous student, Allison Denny, on the gastric cancer pathway in a species of softshell clam, Maya arenaria. We had the very generous contribution of the donated sequence from Charles W. Walker at UNH, who sequenced the genome, and also financial contribution from Maine Inbree, who funded the transcriptome. So our research was conducted on the soft-shell clam Maya arenaria, which is native to both the Atlantic and Pacific coasts of North America. This clam serves a very important role in the New England aquatic ecosystem, but also in supporting the economy by providing a means of commerce through selling it for food and for other means as well. The reason that this species was so recently studied is because there have been increases in cancer occurrences, which are suspected to have been spread through means of horizontal infection. The specific cancer pathway upstream of the gene responsible for the proliferation of this cancer is the RAS-RAF-MEK-ERK pathway, which is a series of signaling hormones and proteins that occur in a pathway that lead to the generation of cancer within this organism. The reason that this research is so profound is that it also could provide a basis for future research into analyzing and better understanding cancer pathways in humans as well. And by providing an opportunity to target a specific pathway to combat this cancer, while hopefully aiming to reduce the cancer and aid in environmental conservation of this very important species. Now, the methods that we utilized in this research include a series of bioinformatical studies using the programs Galaxy, Genome Browser, and NCI Blast, which sound like made up words, but they are bioinformatical computer programs that help quickly process and annotate the genome, which is a series of thousands of sequences that code for the interpretation of different proteins, which are basic genomic directions which order the cell to produce or perform different functions within the organism. And these sequences that we identified in the genome were translated into proteins that we could better understand how this pathway is occurring and how and basically why it's causing cancer. Additionally, because the research conducted on this species is not as well documented as many others, we used other comparative organisms like Crustostrea gigas, which is a Pacific oyster, to better annotate this genome because there were some alternative splicing that occurred and that did not allow for the complete genomic annotation of the softshell clam. So what we obtained from our research was that a series of specific sequences called contags, which are protein sequences that lead to specific genes that are causing the pathway leading to the cancer in the softshell clam. As I discussed previously, alternative splicing occurred in this genome, making it a little bit difficult to understand the complete pathway, and therefore we needed to substantiate those gaps essentially with the Crustostrea gigas genome. But additionally, we were able to provide about a 73% identity to homologous organisms that allowed for filling of those gaps essentially. So what this means is that we are able to publish our findings that can serve as a genomic stepping stone for future research. So by doing the groundwork for other researchers, we have created a means of jumping off and taking more advanced programs to further dive down deep into this genome and annotate it for the understanding of this pathway into a deeper level. Now this does have profound effects, as I discussed previously, to serve in future research for not only the softshell clam and its environmental conservation as it does serve a very important role in the aquatic ecosystem, but also for understanding cancer in general, which the pathway I mentioned earlier, the RAS, MEK, ERK pathway, that one protein RAS is involved in about 30% of human cancers. So that is a very large percentage and affecting millions of people. So if we could do more research specifically on that pathway, then we could possibly find a better target for medication, prevention, and treatment of cancer in humans. So this is a continually explored pathway, and by no means is our research complete, but it is a long coming, and it is a really exciting opportunity to have been able to work on this and to be published in this way for such a profound research project. It was a pleasure to be a part of, and I'm looking forward to see what the future research holds for this.